welcome everybody for this session of Coffee with Joe B at AGBT 2024. It is February 6th. That is 6,000 Plex all day, 6K all day today. I think Arutha was the first person I ever heard say that, and I think it was at last year's AGBT, 6K all day today. So it's great to have you guys here this morning. You guys are leaders in the spatial biology field, so it's great to have you here. You know, one thing I noticed is that each of you come from very different backgrounds and different problems you're approaching. Maybe we can start this off by just giving us an introduction, where you're at, your background, what problem you're really trying to solve, and we'll just work our way through it.
Yeah, absolutely. Right? Great. Thanks for the introduction, Joe. I'm from Brisbane, Australia, so very far away. It took me about a day to get in, so I might be a little bit jet lagged, but I should be okay.
Not you, not you.
Yeah. So my lab started in 2018, deep dive into spatial. GeoMx was our first instrument. Ran 3 samples, got some incredibly beautiful data, and that sort of was that linchpin. We were like, hold on, this is really cool stuff. And so molecular biology background, and what we're trying to do is understand every cancer patient's tumor, right? How do we do a deep dive on response and resistance to therapy? And so the lab's been built out using spatial technologies, and we run head and neck, lung, skin, every tumor, solid tumor we can get on these platforms. And every project is discovery. It's you know, what can we discover in these well-annotated clinical cohorts where we have really good response data, clinical data, and good endpoints.
Hey, it's fantastic. That's why we made these instruments. So great, Arutha. Thanks. Holger.
I had a shorter trip, so I came all the way from Barcelona. So I run a single-cell genomics lab there for the last 10 years almost. So starting in plates, then in droplets, and you name it. But really scaling single-cell sequencing from like 100 cells to 100,000 now. But the thing is, like 3 years ago, we switched to spatial technologies, and that's really single-cell genomics shines in the end, but not dissociating cells, not putting cells kind of isolated in droplets, but keeping them in the environment. So we're like super excited, especially when the 1,000 Plex panel came out. That's where we really came from the sequencing and showed interest in the imaging analysis, and now we are all in. So we tested the whole transcriptome panel. It's just amazing data, and I can't go back anymore. So. No more single cell.
No more single cell. Thanks, Holger.
You just got your next saying.
So I am a translational neuroscientist, and in my lab, we are trying to understand how aging influences neurodegenerative diseases, primarily Alzheimer's disease. So I would say even starting really early in my training, we've always done spatial, but it's been very low plex, just general histology. And in neuroscience, we really rely on that. And so learning about this new spatial high plex world, GeoMx was one of the first instruments my lab bought as well. We had done an experiment with nanostring through the TAP program when I was still a postdoc and got data from postmortem human Alzheimer's brain. The data we got from that was not only were we able to publish, but I was able to get funding from the NIH and the VA to expand my program, and we've now bought a CosMx and doing 6K whole transcriptome as well. And really what shines most for us, though, is the protein data.
Yeah, oh, super. We're going to go deeper into that. So that's a great start. Thanks, Miranda. And, you know, and to just build on that a little bit, I like that. Okay, just imagine your research works. Every experiment you do works. You get funded just at the level you want. 5 years from now, 10 years from now, what is the problem? What is the problem you are going to answer? Don't be bashful. If you want to cure cancer, you should say cure cancer. What is the thing in your dreams you want to make happen in the next 5 or 10 years? Or what's up?
Yeah, no, it's a really good question, Joe. We want to be able to personalize individual therapies based on individual patients' tumors. And so the oncology community, the pathology community is super excited by these because they're getting a new lens on biology and what is driving resistance and sensitivity to therapies, be that ADCs, immunotherapies, whatever your targeted therapies are. And so it's giving us a new lens on biology that's deciphering clinical endpoints right? We can see resistance on the tissue for the first time. We can see sensitivity to therapy for the first time. And even with new drugs, ADCs and so on, we're still using old school chemotherapies, but delivering it a lot better. And so I think, in 5 to 10 years, patient comes in, we get a tumor sample, we run it through the spatial pipeline, and we have an output, a score, a spatial signature that tells you whether the drug works or not before the patient's given it. I think that we have roadmap for that. We can see it's working, and I think that's going to outperform tumor mutation burden, PD-L1 expression, because those are static biomarkers that we're looking at currently. We pivoted during COVID and looked at, we were developing all these tools, these methods, and we started looking at the impact of COVID on the lungs, hearts, brains, and placentas, and we've published across all these studies now, and it's incredible. Like, you can localize where the virus is, we can understand the biology of COVID, the SARS-CoV-2 in the lungs, and the impact of this. And in a cardiac study, we found that, and I'm just saying this because this is the power of spatial, we identified that when we contrasted COVID, the hearts of COVID patients to flu, there was a DNA damage signature in the COVID-19 patients. And so there's a lot of new biology we're learning. And so we can start to marry these up. And so look at the impact of COVID and lung cancer, for example, right? What is the role of infection in cancer and so on? So there's a lot of new biology we're learning.
Hey, super exciting. I love the placenta one that you're just publishing or whatever. It came out a couple of days ago. So yeah, Holger, what do you dream in 5 years, 10 years? What are you going to be?
It's basically what Arutha said, but I think we can kind of frame it more broadly that in the end, what we want to do is digitalize pathology, how pathology assessments are done, right? So you have an H&E stain and you augment 18,000 genes on top. And then you build models for specific diseases as kind of universal diagnostic platform, but then you can model towards prediction of outcome, prediction of treatment response. I mean, in the immuno-oncology area where we are working on, we're looking now at immune cells, infiltration patterns, but then you can augment the cell states, exhaustion patterns, eventually amplification of specific cell types. And that all together, I think, will bring us from looking at an H&E and looking at patterns, but really kind of to learn signatures that are presented by cells, and then they're kind of extracted using the full transcriptome information. That's one. And then we do a lot of work in the Human Cell Atlas project. And then in the end, what you want to do, you don't want to dissociate a tissue, but you want to kind of digitalize it. And that's what we can do now. We just digitalize tissues, organs, organisms. And that's then your kind of blueprint, your map, to map disease tissues on top to find cells of origin of diseases, interaction patterns, and then in the end, Achilles heels for treatment. Yeah. Okay.
So you're skipping the single cell side, dissociated single cell side. You're skipping that part, just going right to the spatial atlasing.
Why would you do that?
Yep, I'm with you on that. Yep.
Could I just make one comment, Joe? I think to Holger's point, we want, we're running all these spatial proteomics, transcriptomics assays. The omics cloud is building, and what we want to do is use that data to impute into an H&E what that looks like. And then using AI, I think in the future, that's, if we can get read of your spatial data on the tissue, on H&E, that's going to democratize spatial globally, right? So if we look at pathology communities around the world, in Australia, we have one pathologist per 100 patients, right? In some countries in Africa, we have one pathologist per country. If we figure out how to use the spatial information to impute into an H&E what that looks like, an H&E stain costs 50 cents. And if you can upload that, run it through some type of algorithm and get your, not just your diagnostic information, but prognostic information, that's really powerful. So I think spatial democratizes digital pathology globally. So I think that's where this will head.
Wow, super exciting. We got Lynn Peterson here taking notes. Do you hear all this stuff? This is the future of medicine here. So Lynn, we've known each other for 15 years, and you write for Trends in Medicine, right?
I would ask you to speak up just a little bit. Joe, you do.
Okay. Speak up a little bit. Okay, go ahead.
So
Miranda.
Where I'm thinking that our program will be in 5 years, and then I will say even beyond. So we have a lot of funded projects right now that last for 5 years, so I'll give you a little taste of what that is, and then think even bigger for maybe 10, 15 years. So we are studying a rare cell. They're called senescent cells. These cells accumulate throughout our body as we get older, And we have found about 2% of the cells become senescent in Alzheimer's disease. These cells have very complex signatures. They're very challenging to find. We have identified one protein marker, and now using spatial, we can use that morphology marker, zoom in on these cells, and look at the transcriptomic profile. And then we take that back to the lab, do different manipulations in mice to try to induce this phenotype, and then what we're doing is treating it. So these senescent cells contribute to many age-associated diseases. Alzheimer's is above the neck, but below the neck, you name the disease, these senescent cells are contributing. And the field of pharmacological interventions to target these cells is expanding exponentially. There are so many companies trying to identify these different therapies for these cells. So now what we're doing is we treat mice with drugs to clear these cells, and we're comparing head-to-head treatments. So we're using GeoMx, and soon we will use CosMx for really high-resolution target engagement. So typically target engagement, you want to make sure your drug is hitting your protein or your pathway, but there's really less known about what it's doing to other cells. And so now using GeoMx, when we treat a mouse, we are seeing not only what happens to that senescent cell, but all of the neighboring cells around it and the whole brain as a whole. And then using that, moving it back to the clinic. So we do have a clinical trial where we're testing one of these agents, but we think that there will be new ones coming down the pipeline. So those are some 5-year goals, but the brain is so complicated. All of the organs that we study in cancer is very complicated. We really want to know how neurons and other brain cells are interacting with one another. So we have a 5-micron tissue section that we're running either on GeoMx or CosMx, but that is a tiny, tiny little snapshot of what's happening in the brain. So we want to apply whole brain or whole tissue imaging, and we're working to put the CosMx and GeoMx data back into that whole brain. So using tissue clearing methods, using light sheet microscopy, and trying to see what's happening at the whole organ. So that's what I think we're working towards for 10 years from now.
Super exciting, super exciting.
I mean, the good news is that the technology is there already. No? So we're talking about digitalizing organs, tissues, but the tech is there. It's just us using it now. So it's not science fiction anymore we're talking about, right?
Yeah, I agree. Hey, thanks for these really inspiring answers. Super neat for me to hear. I'm going to get a little technical on you guys. Many of you actually use multiomic approaches, protein plus RNA. And I'm curious about two things, the importance of Plex in both the RNA side and the protein side, and then also how they work together. Do they generate deeper problems that you can examine, broaden your outlook on how you examine these tissues? So, how does multiomics work for you, and then how does it help you in solving the problems you want to study?
But I can start with the Plex aspect.
Yeah,
so
okay.
I think going from few Plex, let's say 100 to 1,000 to 10,000 to 18,000, it really brings a technology from a targeted, let's say, hypothesis-driven approach to a discovery technology where you can just do a data-driven strategy as we've done for single cell, but that's now completely in space. So we don't come in with a strong hypothesis, but look at the data and then maybe bring it later to a smaller Plex panel for validation. But I think that's where the two ends of the spectrum shines, with more discovery phases or validation. For the proteins, I'm particularly excited using that in the context of immuno-oncology, to look at surface markers, to look at targets, druggable targets, and then to follow them up on treatment in biopsies. So we're doing that now on single cell level, but obviously that is something you want to do on spatial, and then combining the transcriptome with the proteome, it's the best you can do. Yeah.
So our lab, Joe, is very translational, clinically focused, and so we focus on the proteins a lot, and so it's closer to pathology, it's closer, when you show a pathologist transcript data, they look at you a bit confused, and they're like, okay, show me an H&E, show me some protein data, I get, and we sometimes falsely color protein data as dab stains just so that they're happy, they can see brown and white. And then, this is great staining, but we've just flipped the colors. But that's the challenge for us in digitizing pathology. But I think we use the GeoMx is a workhorse. We do 50 to 100 samples a week, just clinical trial samples running through. And the value of that is we have good clinical endpoints, so response, overall survival, progression-free survival, and so on. And we have cohorts where we have good responders and maybe poor responders. We can now, using the IPA and some of the protein panels, we can discover hundreds of markers that may be, differentially expressed between the groups, and that gives us a lens that we can then go validate. The protein's important because it lets us build panels and smaller panels that we can move to the clinic. The RNA is important because we can start to understand mechanisms of resistance and so on. And so we recently ran a trial where we were looking at a clinical trial cohort from pharma, and we identified the mechanism of action using whole transcriptome data, and then the protein data told us why the T cells weren't infiltrating that tumor. And so there's really nice biology when you overlay them and integrate them, and hopefully, same slide, that's where the power is. We lead with protein, but we need to use the RNA to understand sort of functions, mechanisms, and those sorts of assessments.
That's great. There's so many people leading with RNA. It's nice, to be honest with you, it's nice to see you leading from the other direction. I think they're both super important. Yeah, go ahead, Miranda.
So for these senescent cells that I was talking about, the multiomic approach really is where the field is with senescence biology, a necessity. So the gene signatures were described in these senescent cells, but the proteins are upregulated, but most of the antibodies are really terrible. And so we really do rely on the gene expression, and these senescent cells, their signatures are very different depending on what cell type became senescent, what stressor caused the cell to become senescent. So we really do need a very large transcriptomic panel to identify them. But then, like Arutha said, when you target them or you develop drugs for them, we need the protein signatures. And so we started with a gene signature, identified a gene that was upregulated, found that the protein was also upregulated. Now we're using that protein as our morphology marker, and then continuing to cycle through it and get more data on the transcript.
I guess one other thing I'll ask you, when you study Alzheimer's and stuff, you have protein signatures you look at, like all the phospho-taus, that seem to me absolutely essential for understanding progression of disease in the brain.
Yeah, absolutely. So all of the neuropathology in Alzheimer's disease is protein-based. And one of the hallmarks, these neurons, they're called neurofibrillary tangles. They have really high levels of phosphorylated tau, which is a microtubule-associated protein. But tau can become phosphorylated on over 20 epitopes. And historically, it has just been sort of a yes/no. Is there tau phosphorylation? Do these have tangles? But now with the GeoMx panel and even CosMx panel, there's five different taus in there, phospho-taus. And now what we're seeing is that these tangles, it's not all just tons of phospho-tau. The phospho-tau is localized differently in different areas of the cell, in different cell layers of the brain. So we're now finally starting to understand a little bit more about the biology. It's not just, one phospho-tau equals a tangle.
Yeah, no, super, super interesting and super important. It's not just RNA and protein, but it's the phosphoproteins and all the other post-translational modifications that are on there, and we need to get better and better at doing that. I guess experimental design and spatial, you're spatial biology leaders, and then you got to be thinking of experimental designs. I know you all have experimental approaches. Maybe just describe how you lay the path, the roadmap for you to succeed. I guess we've talked about multiomics. What are the other tools you think are really important to be bringing into play? They can even be ancillary things that aren't spatial things, other important things that feed in.
So one of the questions that I get a lot when I present my data, and it's a question that we think about a lot in the lab, is how closely aligned histologically are these brain samples? So one of our projects is evaluating when in the adult human lifespan we first see these senescent cells, when does Alzheimer's pathology first appear? And so we're looking at over 200 individual cases, so 200 different people's brains, and we want to make conclusions about them. And so one question I get all the time is, are these brains from the same exact brain region? And the short answer is no. And so we think a lot about how much does this matter. With mice, we have a lot more control over that. So we're really trying to answer that question. And understanding how many tissue sections you need from each animal to make that conclusion. So that goes into the experimental design. But the scan area is actually pretty big on both the GeoMx and CosMx. So we're able to put 12 mouse brains on one of those slides and directly compare across treatment groups or multiple sections from that mice. If you plan the experiments well, and when we are starting collaborations, we say, really, if you plan the experiment well, you might only need one slide, which is pretty remarkable.
Yeah, no, I like that. Yeah.
I think we're very excited about these longitudinal aspects of sampling, where you have material from patients before and after treatment and following them up on treatment. So we work a lot in immuno-oncology. So we have single-cell data sets where with biopsies following up patients on checkpoint inhibition, in colorectal cancer, patients that respond or do not respond. And actually on single-cell level, they look quite similar. So there's a lot of activity ongoing, also non-responders, a lot of new infiltration of T cells. And we can't really understand why then the tumor is not cleared, right? So in the MSI high patients, tumors are, even with the neoadjuvant therapy, are kind of cleared without surgery even. And in MSS tumors, it's not the case, but the activity of immune cells that we see is enormous. So now we need this spatial information to know why the cells are not killing, although they're infiltrating, or they're going into the tumor, but they're not actively killing tumor cells, right? And that's the spatial information I think you need.
You know, another one that you were mentioned to me before we started this interview was that you have these samples where you can see different almost time progressions in a single sample.
Yeah.
And I think that's really fascinating because they're there in space and you can see, describe a little bit what you were talking to me about this sort of, you can see these different time series in the same tumor sample, sort of N=1 kind of.
But then I'm spoiling what I'm presenting tonight.
Ah, okay. But no one's going to hear this until later. When does this come out, Kevin? Okay.
So for the whole,
okay, but just give them a teaser.
Yeah, so for the whole transcriptome, we profiled polyps, which are very specific type of polyps that can give rise to classical adenocarcinoma, colorectal cancers, but also to mucinous carcinomas. That's the form that is more responsive to immunotherapy. And the polyps that we're analyzing, so they're called TVAs, are very specific subtypes. So within the cell mass, you have cells of origin of both cancer types. So somehow the tumor at one point, or these neoplastic pre-lesion decides at one point if it wants to be more aggressive, or there's some selection, kind of evolution that we now try to understand looking at single cases. So within a case, we have the normal, we have different diseases, different areas of progressive diseases, even tumors sometimes inside. So within a single case, we can then looking at 500,000 cells from one slide, right? We can just map out everything, which kind of is the entire evolution of the tumor. And we can start figuring out why or when the decision-making is done, being a more aggressive, less aggressive, better treatable or not tumor.
Then you anticipate doing like RNA trajectory analysis on these things? Would they be useful for you, Holger, or that will be
I think you can leverage a lot of tools that
Okay.
we've been using for the single cell analysis. So actually the normalization that we've done now on the whole transcriptome is the same that we're doing for single cell. So we can do library size, kind of the same transformation of data because we have full transcriptome information. And then you can leverage all the tools. But I think the sweet spot is not to put cells again into UMAP, but to never use UMAPs again, right? To work directly in the spatial
Yeah,
context.
there's a challenge for you. Yeah, no, I love it.
Yeah, just to add to Holger's point, we're now looking at longitudinal, we're calling it a spatiotemporal study, where we're looking at longitudinal skin cancers. In Australia, melanoma is one of the biggest killers, and so we're looking at patients that progress from sort of early stage nevi all the way through to full-blown melanoma, but we're getting skin biopsies at every 6 weeks on the same site. And so we're getting tissues at cure and at progression. And so now we're going back and we're mapping the spatial changes over time. The initial time point data looks incredible. it's challenging to analyze the cured patients because you're going from having tumor to having no tumor. And so, but we have those data points, and skin's easily accessible. So initially, spatial was for us single time point, and then we're predicting, but now it's, to Holger's point, we're getting those longitudinal samples, and that's going to really showcase the changes in the microenvironment over time under the stresses of therapy, and then so on, and that's really exciting for us.
It's a unique design for melanoma. Yeah. There's just a handful of diseases like that, but boy, you can learn so much. Could turn it technical again for sort of narrow it down a little bit on something. And it has to do with a thing that we spend a lot of time on inside NanoString is trying to generate really good segmentation patterns we're measuring all this high-plex RNA and protein. We really want to make sure the RNA is going the right bag. And maybe can you describe what you think the role of segmentation is for this whole field of spatial biology? Just
there you go.
I have a quick answer.
Yeah.
So if you can't segment, it's not single cell.
Yeah. Okay.
Because if you want to do spatial single cell sequencing, if you don't do or get the segmentation right, you end up with the mini bugs, bleeding transcripts.
Yeah.
So then the analysis is harder and harder. You then have to rely on computational tools that clean the data for you, but that's much harder. So we see that from the single cell context as well. If we have ambient RNA, so that's kind of noise you introduce in the data that's very hard to get rid of later. So you need to get the segmentation right for the spatial. And then you do real single cell analysis in space.
Yeah. No,
that's a great answer. Super simple. If you don't segment well, you're not doing single cell biology anymore.
No. It's critical, Joe. I mean, we spend days trying to figure this out. There was a session last year at the American Association for Cancer Research meeting, a 3-hour session on just cell seg. And so it's one of the biggest challenges for the field, but the time and the effort that NanoString spent in improving the cell seg has been, it's been incredible. We've seen other sort of approaches where you do, cell expansion and so on. It doesn't work. And so when you go back and you compare the data, it just, cell seg is critical, and we need good cell seg and iterative versions of it. Yeah. And maybe we can improve it by looking at the 3D space and incorporating that dimension. But ultimately, you need really good cell seg. It's the hardest tool, I think, the hardest challenge for spatial today, a really good cell seg. Yeah. It's critical.
And I guess, Miranda, the brain is the biggest challenge of them all, I think, for segmentation. But
yeah, I mean, I would just echo and amplify what they're saying, because you have all of these cells in the brain, and they have projections that can go really, really far. The neurons talk to each other across the brain, and you have all of the glia interacting with them. And if you don't have correct segmentation, like you said, you're not doing single cell anymore.
Yeah. So this is, I got a question for Arutha and Miranda, okay? Because I think you both have actually performed both what you call profiling with the GeoMx Digital Spatial Profiler, and you've also performed spatial molecular imaging with CosMx. Can you describe in general how these two different ways of examining spatially tissue, how do they work together and synergize or not? It's up to, take a pick. Yeah.
So something that Arutha said earlier is the GeoMx is the workhorse. And in our lab, it's the same. The GeoMx is really the workhorse. We can scan a lot of tissues, and that's really more, for us, hypothesis-driven. So we carefully select our morphology markers to really zoom in to specific cells or regions we want to look at. Whereas CosMx, you get an entire image of the whole brain. And with those data, some of the really cool analyses that have been impactful for us are the spatial niches and looking at how these change with disease progression or with age. Also identifying rare cells, which, with GeoMx, if we are already targeting which cells we want to look at, identifying rare cells is far more challenging. But with the CosMx, we've identified some rare cells and looking at how those rare cells are impacting their environment. So we go back and forth between GeoMx and CosMx a lot. The data are very complementary, but we use them slightly different, hypothesis-driven versus discovery.
I might give you an example, Joe. So during COVID, we were looking at the lungs of COVID-19 patients at autopsy, and so we went to the pathologist with these H&Es, and so we said, what are the interesting structures? Here's the RNA overlay of where the virus is, and so they'd annotate the bronchioles, the alveolar structures, all the really interesting histology on the tissue, and so we knew that using GeoMx we can go draw around those structures, liberate the whole transcriptome, and we can figure out from a discovery lens, we can contrast between COVID and flu, COVID and normal lungs, and so on. It is limited in resolution, but it gives us structural information that's pathology interpretable, but also important for the pathology community. So we discovered this type 1 interferon, which was highly enriched for in COVID-19. So we knew this was specific to COVID-19. We knew what structures it was in, and now we wanted to go in and figure out, hold on, what cells are they in? And so we went from the GeoMx through to the CosMx, and we just had the paper accepted in eBiomedicine. It'll come out next week. We could localize that IFI27 to a specific subset of macrophages using the CosMx. And so for us, it's complementary. We're thinking of it as, pathology, structural data with GeoMx, high throughput, and then narrow down into a really focused question with the CosMx. And I think as the CosMx plex and throughput improves, then we might move in a certain direction. But right now, it's a GeoMx CosMx stack for us. Yeah.
That's great. Thank you. Okay, I got one for you, Holger. And I don't want to take any of your thunder from your talk tonight, so you could just talk in generalities. I think you're probably the very first person in the world that's looked at the CosMx whole transcriptome and then have a controlled serial section underneath that where you can actually look at chromium. I think you used the Plex kit on an FFPE, and then you got this data right on top of each other. And you're a single cell expert from the beginning. What's your impressions of the CosMx whole transcriptome when viewed in that light?
It's now fully spoiling my talk.
We can make sure they don't put that part out in the public. They're not going to.
In essence, it looks the same. So we've done as you said, we have serial sections. We've isolated nuclei, the PathoSeq protocol. We've analyzed 40,000 cells per sample on the Plex. Sequence quite deep to 20,000 reads per cell. Yep. So kind of the cost between, I guess, a CosMx run and this Plex run is more or less the same, I guess, around like $5,000 euros. But then you get 10 times more cells on the CosMx. So 500,000 cells against 40, 50,000. And so that's
night and day.
Yeah, it's crazy. Yeah. And then when you count transcripts per cell or genes per cell, it looks the same, looking at the epithelial fraction. So for us, the 80% were epithelial cells. The single cell looks slightly better for the immune compartment, but still with the CosMx data, we get around like 400, 500 genes per cell for immune cells, and that's crazily high with spatial information, obviously. So it allows us to zoom very deeply into the epithelial compartment. So we have subclusters for healthy colon, subclusters for TVA, so these transfer polyps. We have subclusters for the cancer part, and it's all we can align in trajectories if we want. But it's just giving you this full high resolution of a single case, and you have the full transcriptome information underneath. So it's not selecting single markers, it's just you have the entire signature of a subcluster. And this is what it kind of leads you then through the interpretation of the evolution of that single case. But again, it's a single case, it's crazy. You get so much information from one sample, and then if you kind of scale that across cohorts, across kind of treatment scenarios, it's just information that we can't really imagine where we're going. Yeah.
I think that's super neat for us because, I remember sitting in a research room on a whiteboard saying, can we do this? Can we dream and do this? And to see it come out for real is, so for all the R&D people watching the video, the huge thanks. And it's really neat to see that come together.
And if it's the same cost, so why would you go back, right? So why would you dissociate again? Mm-hmm. So you get 10
no
times more information.
Yeah.
And then in the end, if you do the subcluster or substructure analysis, the number of cells has an impact. So even though you would detect slightly less genes or transcripts, if you have 10 times more cells, you can do very much the same or even more. Yeah. Plus, you have the spatial information. Plus, you don't dissociate, so there's no artifacts involved. Plus, and so on and so forth. Yeah.
Hey, super exciting. I think put you to enough torture test here, but I would like to open it up to anybody in the audience that would like to have any question for our panelists here today. And it's open.
do one.
Yes.
So with the whole transcriptome on CosMx, we say we've delivered sort of the holy grail of spatial. What do you think is next? Once we've achieved that and delivered that in 2025, what should we focus on next?
Okay, is that loud enough to hear, Kevin, you think? Okay, we got it? Okay, go
I have
ahead.
an answer. So did we get the question? Do I need to repeat it?
No, I think
Okay.
it sounds like we got it, so that's the answer. Yep.
Splice
Fred's got a nice
variants.
booming voice. So.
Slice variants.
That's super important so my favorite gene and protein in life, tau, there are over 30 splice variants. 6 are predominantly expressed in the brain. So right now we have information on tau gene expression generally, multiple phospho-taus, but I want to know the splice variants.
Okay, that's a good one. We just recently made a custom 300-plex panel for isoforms in the brain. So, and they actually all succeeded. It was really nice. We designed them and it worked. So you should really talk to our custom people. If you know what the isoforms are at the sequence level, we should make them.
They're so similar. It's challenging, but
Okay.
I hope you guys can help me with this.
Okay, yeah, bring the design in. Love to give it a try.
It's an interesting question, what comes after the Holy Grail. No? So I mean, we're excited to use then also spatial as a tracking tool, and then looking or augmenting information like TCR sequencing on top, so we can have a lineage tracing tool that allows us to quantify amplification or activity of T cells in space, and so kind of to track them then over time. I think that's technically more challenging, and we had the discussion, but I think that's something useful to augment, especially in the immuno-oncology context.
Yeah, Brad, I never thought it would happen this quickly, but it's incredible to see whole transcriptome, single cell, and subcellular too. I mean, it's a incredible achievement that, I remember the days where Joe would, talk about 96-plex RNA on GeoMx, and then you come for AGBT, it's 1800, and then whole transcriptome, and you've done it again for CosMx, so incredible. When the announcement came out, nothing made me happier that day. I was just like, that's like, you've hit Everest there, and then that's kudos to the lab and everyone behind and the R&D team behind that. For us, we're excited about using that, and it goes back to the spatial cloud of information, this omic data set that sits above your H&E, and so we're doing a lot of work trying to impute that into the H&E and get pathology interpretable features that we can go and say, this is not a black box readout that is associated with your clinical endpoint. It's an actual, it's a feature that you can see and measure. And so that's what we're hoping to do with the whole transcriptome, integrate that into a multiomic analysis, and then align that with the H&Es, and then get the pathology community involved, oncology community involved behind it. It's all moving into this digital pathology space, and absolutely blown away by, I can't wait for Holger's talk tonight.
I have a question. So with all this plexing, fantastic, you generate a vast amount of data, including all these images, and it can be a challenge. So how do you see data analysis in general for spatial evolving? How do people deal with it? What do you feel is the best way forward?
Hear that one, Kevin, you think? Okay, we can answer right away. Go ahead.
I think it's, as I said, you can use tools that were designed for single-cell analysis now. And there are many, there are thousands of tools now. But it also gives us the chance to develop new tools that appreciate now the full transcriptome, as we have from the single cell, but then also the spatial context. Plus, we have now millions of cells. We just published a tonsil atlas with around 500,000 cells. It took us 3 years and another year to publish. And now it's basically one run on the CosMx, right? So we have all that information. So you don't probably, if you look at healthy samples.
Is that an immunity cell every year?
Yeah. That came out last year, last week. So it's really the scale, I think, that makes it also more challenging. So the tools have to cope with larger cell numbers, more data, but also they can then appreciate more cells, the spatial context combined with the full transcriptome, and probably combine tools that we use already for the single cell and kind of bring them to the next level, which is then space. Yeah.
I can take it. So I remember running one of the first CosMx runs, and the initial data sets, this was pre-atomics, was about 10 terabytes of data off one slide. And I went to one of the data guys in the lab, and I'm like, why isn't this opening? And like I came back half an hour later, and it was the same. I was like, oh God, here we go. But we've in getting these, but they're incredibly rich, right, Joe? They're incredibly rich data sets. We're not looking at 20, 30, 40,000 cells. We're looking at millions of cells in space, X, Y, and Z coordinates, and we haven't even integrated the Z component with a lot of these. We're sort of collapsing it to analyze it. So I think there's such a rich data set there, and one of the new data guys in the lab, he built his own keyboard on day one, and I was like, okay, he's good. And so all he wants to do is use the supercomputers at the university and modularize the analysis pipeline. So I think, to Holger's point, we're going to start incorporating a lot of these single cell approaches, but also developing custom spatial analysis tools. And I'm excited by how we can then integrate that into atomics, because it's very open-ended in that form. So I think it modularizes over time, becomes easier to analyze these data sets. A lot of cloud-based solutions will come through, and that's going to be the power of really democratizing it, being able to sit and have a collaborator in Spain or the US, and we're looking at the same image and analyzing it at the same time, because you can't have that locally, and you can't be sending hard drives around the world.
Absolutely.
So I have a question. We've discussed achieving the holy grail of transcriptomics, but where do you dream of going with proteomics, the spatial?
It's the holy grail, proteomics, spatial.
Yes, please, all of them.
AGBT 25.
Yeah. All of them, post-translational modifications, clean fish break, everything, yeah. But I mean, already what we have with the nanostring panels are incredible. I never thought that I would be able to see all of these unique phosphotau's within a cell. So the primary antibodies are all raised in the same species. And so it's like, you do serial sections and choose one, but now we are seeing them all and seeing how differentially localized they are. So it's so informative. But yeah, we're scientists. We always want more. And then when we get more, then we get the question, how are you going to analyze the data? And then we have crises and then move forward.
Good one.
Yeah, I think that the proteomics part will give more insight into the functionality of cells. So with the RNA, basically what we do is phenotyping, right? But with the ATAC-seq data, you can do mechanisms. With the proteome, I think you can do much better job on functionality.
I think we could. Yeah.
And we heard a lot of medical things here in this interview today. Lynn, you have any question you would like to ask from your trends in medicine perspective?
How fast is this going to translate actually into treatment? Even if somebody invents a new drug, it takes forever to get it, in most cases, to be applied to actual patients.
Think that picked up on the auto, Kevin? Okay, you're all right. This is a great question, and I think it's a very important one. It always happens too slow, but maybe Arutha, you kick us off, because you really pushed the envelope in this direction. How fast can you make this thing happen?
Yeah. Lynn, I think it's a great question. We just saw the world's first spatial score being announced last year, which is FDA approved based on a 9-plex spatial signature. So 9 markers define this DNA damage signature, and that can be used as a companion diagnostic assay. And so if we do high-plex protein discovery work on clinical cohorts where we have good endpoints to measure, we can figure out what are the markers that define resistance and response. And then you could build that into a panel that you can go and screen in a high throughput manner. So I think spatial is the only technology that gives you that. If you look at other measurements, genomic measurements, tumor mutation burden, histology measurements like PD-L1 expression, they're very limited and they don't associate with endpoints. TMB is a great example of, just dropping the TMB as to what a high TMB is was just to include more patients in the trial. The actual benefit of giving, what is a high TMB today? No one knows, right? Is it 20 mutations per megabase? Is it 10? And so having a high TMB meant that you have more neoantigens, the immune system will recognize those neoantigens, and the immunotherapy will work better. But the drug companies kept dropping what a TMB was just to increase the number of patients being given drug. And so the actual clinical benefit was sort of mixed. And so we need better assessments of the tumor microenvironment, and I think we now have the tools and we have the discovery tools to be able to figure out what are the markers we need to, that will define those endpoints. There is a path we can see that. There are FDA-approved assays now on the market. So I'm really excited by that. This next 5 years, I think we will have spatial signatures in the translational and clinical space.
5 years.
So we are using repurposed drugs, so that helps expedite the translational path. So one of them that we're testing in clinical trials right now is a repurposed chemotherapy along with a flavonoid for Alzheimer's disease. But also an area that our lab is really interested in developing is spatial on microscope slides. So having cells on a slide and treating those cells, because we know even if there is a supposed homogeneous cell population and you treat them with drugs, not all of the cells respond the same, and we have really no idea why. But with these spatial platforms, we can now start to answer that. So I think that's going to be incredibly informative.
Yeah, that's a very interesting thing. We actually didn't talk about that in this interview, how you can actually do ex vivo type of things. You can do co-culture, organoids, all sorts of things, drug treatments, and now spatially probe those fixed cells. And the transcripts go through the roof, right? Over 10,000 transcripts per single cell with the whole transcriptome looking at those fixed cells. So yeah, the amazing amount of information you can get out of that. That's a good one. Go ahead, Holger.
I agree with what Arutha said. So I think the lowest hanging fruit is probably IO, immunotherapy prediction, because we have markers that are very poor in predicting outcome. But in the end, we have a therapy that is targeting immune cells, but most of the markers are on the tumor compartment. So if we now look at immune cells in space, I think we find, not easily, but we find kind of very predictive or good markers that from one section before treatment will allow us to predict the outcome and in the end, the response to IO.
Okay, I have a follow-up.
Okay, say it nice and loud, so
Okay.
if you don't mind.
So the other area that we're looking at in oncology is ctDNA. And right now, one of the issues is, okay, so we can look at this and say a person is going to probably recur, but we don't know if treating them is going to make a difference, or if the drugs we currently have will work in a recurrence that's found by ctDNA, not found and analyzed differently. How can this help with that? Because ctDNA is really a, something everyone wants.
Yeah. So I did my PhD in the liquid biopsy space, so it's very close to my heart. I think ctDNA is a great assay in the advanced setting. It's not really going to help early stage, the stage 1, stage 2 disease. You can look at all the GRAIL data. It's limited, and so it's great for tracking mutations over time, and but you need to know what you're looking for. So we need to go back to the tumor, and I think that's where it starts. The reason I shifted from ctDNA research to tumor is because we're not capturing the heterogeneity, all the mutations that are happening by just looking at the blood alone. We need to match the blood data with the tissue data. So that's why my lab shifted to going and figuring out everything we can from the tumor, including the mutational profile, and then we can track that in the blood. I think ctDNA is a great assay. There's high value in them in longitudinal and sort of monitoring studies. We have patients that increase ctDNA over time, generally associates with a worse outcome, but you need to know what you're tracking. And sometimes you might be tracking the wrong mutation. And so you might have one increase and one decrease, and so what are you tracking over time? So I think you need to match tumor mutational profiling with blood for the same patient, and then that'll be one way you can incorporate ctDNA into it. But I think we need to overlay that with the heterogeneity that we're understanding of the cancer using spatial and hopefully mutational profiling in the future. Yeah.
I have a question for my panelists. Have either of you looked at blood, like a blood smear on a slide with either GeoMx or CosMx?
The answer is yes, we have done that. It's super interesting, actually. I would love to be doing some profiling, and I think there's this whole area that we just call cell-based research that works on both CosMx and GeoMx. It's really totally an untapped avenue, and I think especially as we get these things to the right plex, now both GeoMx and CosMx are at whole transcriptome, so I think that cell-based assays are really ripe for taking hold of.
Yeah. So we've actually started doing this, looking at PBMCs, and so I'm excited by Holger's talk because we'll be able to, we get blood from patients, not smears, but we spread them across multiple slides, but we'll be able to do potentially whole transcriptome and high-plex protein on this at a single cell resolution. And so we can measure the tumor cells, all the immune cells in there. That becomes really powerful, and it's a new application, I think, for applying spatial to blood-based assays.
Yep. And it's very inexpensive. Another thing, like on the CosMx, it's less than 1 cent a cell. So you're getting a whole transcriptome for less than a cent on every one of those cells. It's pretty amazing,
We
actually.
used to have to pick them using a micromanipulator and then go sequence, but now you can get it.
Don't pick them anymore. That's a good one.
That's it. So you have the option again to scale single cell sequencing. So it's for cells in circulation. It's for to scale what we're doing on single cell, but not then in droplets, but on a CosMx run.
Don't need it.
But then
Don't need
in
a
magnitude higher, blowing cell numbers up.
Yeah, I'm very excited about that application area.